Hi guys, welcome to NEET Made Easy. Today we are going to learn about the tools of recombinant DNA technology. First, let us know the key tools required for rDNA technology. Restriction enzymes. These are enzymes used to cut the DNA bands. Polymerase enzymes. These are used for the generation of multiple copies of the rDNA formed. Ligases. These are enzymes which are used to attach the DNA particles cut by the restriction enzymes. Vectors, these are used as the vehicles or transporters to transport the desired gene into the host organisms. Now let us know about the restriction enzymes. First, let us go a bit into its history. These were first discovered in the year 1963. These were discovered while studying the reasons for the restricted growth of bacteriophages in E. coli. They found two reasons for it. The first one was adding of methyl groups, and the second one was an enzyme that was the reason for the cutting of DNA. Later, these were named as restriction endonucleases. Later on, the first restriction endonuclease characterized and isolated was HIND2. It was done 5 years after the discovery of the restriction enzymes. HIND2 cuts DNA molecules by identifying a particular sequence of 6 base pairs. This sequence is known as recognition sequence. The HIND2 cuts the DNA particles at the sequence GAA-TTC. Restriction endonucleases cuts the DNA molecules by identifying a specific palindromic sequence. Palindrome means a word which spells the same when read from right to left or left to right. For example, Malayalam, when it is spelled from right to left, it spells the same as when it is spelled from left to right. As far as known, about today there are about 900 restriction enzymes discovered from about 230 bacterial strains. Now let us learn about the nomenclature of restriction enzymes. Let us learn it by taking an example which makes it simpler. Let us take E. coli 1. The E in this denotes the genus of the bacteria, that is Escherichia. The next two letters, they are species, they denote the species, that is coli. The next letter R, it denotes the strain of the bacteria from which the restriction endonuclease is derived. Next, the Roman letter, this denotes the order of the strain from which the restriction endonuclease has been derived. Now, let us know in which class of enzymes restriction endonucleases have been placed. Restriction endonucleases have been placed in a larger class of nucleases. Nucleases are of two types. They are exonucleases and endonucleases. Exonucleases cuts the DNA at the ends of DNA bands. Endonucleases cuts the DNA at specific sequences within the DNA. Now, let us learn the action of restriction endonuclease. Let us learn it through an example of E. coli 1. E. coli 1 finds the specific sequence of it, that is GAA-TTC. It finds the same in the vector DNA and the foreign DNA which contains the desired gene which needs to be inculcated into the new rDNA technology, rDNA. It cuts between the GA in both vector DNA and foreign DNA. It does not cut at the center of the palindrome. It cuts a bit away from the center, making the ends of DNA sticky ends. These sticky ends easily forms hydrogen bonds with the counterparts of the cut parts of the vector DNA. These sticky ends facilitate the action of the enzyme DNA ligases. Thus, the cut vector DNA and the cut foreign DNA both attaches to form the recombinant DNA. Now, the cut parts found by the action of restriction endonucleases need to be separated and isolated in order to make them in a form that they can be utilized. Uh, the separation and isolation of the cut parts of DNA is done by the process called gel electrophoresis. As we know that DNA is a negatively charged particle, in the process of electrophoresis, DNA particles, to be precise, DNA fragments are separated by forcing them to move towards the anode under an electric field through a matrix. The matrix used for the gel electrophoresis is agarose gel. This is extracted from seaweeds. The separation takes place according to the size, that is, smaller the size of the fragment, farther is it moved. Separation fragments can be only visualized after staining with ethidium bromide, followed by the exposure to UV radiation. On visible light, the separated fragments cannot be seen clearly. On staining with ethidium bromide and exposure to UV radiation, bright orange color bands of DNA is seen. The separated bands of DNA are now are cut and separated from the uh, matrix and extracted. This process is known as elution. These purified DNA bands are then used in the synthesis of rDNA by joining with the vectors. Now, let us see how gel electrophoresis takes place in the diagram. Here we can appreciate wells. The cut fragments of the DNA are kept in these wells, and on application of electric field through the sieving effect, the DNA fragments move towards the anode according to their size. The smallest DNA fragments move the farthest from the well. The largest DNA fragment stays near to the well. Now let us see some MCQs which have been appeared in previous NEET questions in order to improvise our learning. First, DNA fragments are negatively charged, neutral, positively charged, either positive or negative based on the size. Just now we have learned that DNAs are negatively charged particles, thus the option is A. Second question, the DNA fragments separated on agarose gel can be visualized after staining with. We have learned that the DNA fragments which have undergone electrophoresis can only be seen clearly on staining with ethidium bromide and followed by the exposure of UV radiation. Thus, the option correct for this question is C. Third question, the DNA fragments generated by the restriction endonuclease in a chemical reaction can be separated by. We have learned the process of electrophoresis, which is required for isolation and separation of the DNA fragments generated by restriction endonuclease. This concludes our class today. Thank you.